Welcome to Bio 211D Plant Physiology Unit 4, Session 2, Fatty Acid Catabolism. And by the close of this session, students should be able to state the sources of fatty acid, describe the composition of fatty acids, describe the digestion of fatty acids, explain the transport of fatty acids into mitochondria, describe beta oxidation of fatty acids, and name some enzymes involved in digestion, transport, and oxidation of fatty acid. So fatty acids are a family of molecules classified within the lipid macronutrient class. Remember, we said that they also serve as substrates for cellular respiration in the energy production in the form of adenosine triphosphate, that's ATP. When compared with carbohydrates and protein, fatty acids yields the most ATP on an energy per gram basis. ATP is produced by a pathway called beta oxidation. This form of oxidation we'll discuss in detail later. Moreover, fatty acids are important for energy storage, phospholipid membrane formation, and signaling pathways. Fatty acid metabolism consists of catabolic processes that generate energy and primary metabolites from fatty acids, and anabolic processes that create biologically important molecules from fatty acids and other dietary sources. In this session, we shall concentrate on the catabolic processes that release energy in the form of ATP. Fatty acids are stored as triglycerides in an organism. They are an important source of energy because they are both reduced and anhydrous, lack of water or water-free. The energy yield from a gram of fatty acids is approximately 37 kJ compared to 17 kJ per gram for carbohydrates. Since the hydrocarbon portion of fatty acids is hydrophobic, that's water repelling, these molecules can be stored in a relatively anhydrous environment. Digestion and transport of fats. Cells can obtain fatty acid fuels from three sources: fats consumed in diet, fats stored in cells as lipid droplets, and fats synthesized in one organ for export to another. Some species use all three sources under various circumstances. Others use one or two. Vertebrates, for example, obtain fats in the diet, mobilize fats stored in specialized tissue, that's adipose tissue consisting of cells called adipocytes, and in the liver, convert excess dietary carbohydrates to fats for export to other tissues. On the average, 40% or more of the daily energy requirement of humans in highly industrialized countries is supplied by dietary triacylglycerols, although most nutritional guidelines recommend no more than 30% of daily caloric intake from fats. Triacylglycerols provide more than half the energy requirements of some organs, particularly the liver, heart, and resting skeletal muscle. Stored triacylglycerols are virtually the sole source of energy in hibernating animals and migrating birds. Protists obtain fats by consuming organisms lower in the food chain, and some also store fats as systolic lipid droplets. Vascular plants mobilize fats stored in seeds during germination, but do not otherwise depend on fats for energy. Fatty acids are usually ingested as triacylglycerides, which cannot be absorbed by the intestine. Triacylglycerols are the most abundant dietary lipids. They are the form in which humans and other animals stored reduced carbon for energy. Each triacylglycerol has a glycerol backbone to which are esterified 3 fatty acids. Most triacylglycerols are mixed. In other words, the 3 fatty acids differ in chain length and number of double bonds. So you have glycerol and triacylglycerides. So they differ in the chain and the bonds as indicated earlier. Digestion and transport of fats. Lipases hydrolyze triacylglycerols, releasing one fatty acid at a time producing diacylglycerols and eventually glycerol. The glycerol arising from hydrolysis of triacylglycerols is converted to the glycolysis intermediate dihydroxyacetone phosphate by reactions catalyzed by glycerol kinase and glycerol phosphate dehydrogenase. So fatty acids are broken down into free fatty acids and monoglycerides by pancreatic lipase, which forms a 1:1 complex with a protein called colipase, which is necessary for its activity. So the activated complex can work only at a water-fat interface. Therefore, it is essential that fatty acids be emulsified by bile salts for optimal activity of these enzymes. That is, invertebrates, before ingested dried glycerols can be absorbed through the intestinal wall, they must be converted from insoluble macroscopic fat particles to finely dispersed microscopic micelles. This solubilization is carried out by bile salts such as thyrocholic acid, which are synthesized from cholesterol in the liver, stored in the gallbladder, and released into the small intestine after ingestion of a fatty meal. Bile salts are amphipathic compounds that act as biological detergents, converting dietary fats into mixed micelles of bile salts and triacylglycerols. The digestion products of triglycerides are absorbed primarily as fatty acids and 2 monoglycerides, but a small fraction is absorbed as free glycerol and as diglycerides. Once across the intestinal barrier, they are reformed into triglycerides and packaged into chylomicrons or lipoproteins, which are released into the lacteals, the capillaries of the lymph system, and then into the blood. Eventually, they bind to the membranes of hepatocytes, adipocytes, or muscle fibers. In these tissues, they are either stored or oxidized for energy. The liver acts as a major organ for fatty acid treatment, processing chylomicron remnants and liposomes into the various lipoprotein forms, in particular very low density lipoprotein VLDL and low density lipoprotein LDL. Fatty acids synthesized by the liver are converted to triglyceride and transported to the blood as VLDL. In peripheral tissues, lipoprotein lipase digests part of the VLDL into LDL and free fatty acids, which are taken up for metabolism. This is done by the removal of the triglycerides contained in the VLDL, that's very low density lipoproteins, so for metabolism. And this is done by the removal of the triglyceride contained in the VLDL. What is left of the VLDL absorbs cholesterol from other circulating lipoproteins, becoming LDLs. LDL is absorbed via the LDL receptors. This provides a mechanism for absorption of LDL into the cell and for its conversion into free fatty acid, cholesterol, and other components of LDL. The liver controls the concentration of cholesterol in the blood by removing LDL. Another type of lipoprotein known as high-density lipoprotein or HDL collects cholesterol, glycerol, and fatty acid from the blood and transports them to the liver. So in short, chylomicrons carry diet-derived lipids to body cells. VLDLs carry lipids synthesized by the liver to body cells. LDLs carry cholesterol around the body, and HDLs carry cholesterol from the body back to the liver for breakdown and excretion. When blood sugar is low in human beings, decreasing insulin levels signal the adipocytes to activate hormone-sensitive lipase and to convert triglycerides into free fatty acids. These have very low solubility in the blood, typically about 1 microliter. However, the most abundant protein in blood, serum albumin, binds free fatty acid, increasing their effective solubility to about 1 millimole. The Serum albumin transports fatty acids to organs such as muscle and liver for oxidation when blood sugar is low. Neutral lipids are stored in adipocytes as well as in steroid synthesizing cells of the adrenal cortex, ovary, and testes in the form of lipid droplets, with a core of sterol esters and triacylglycerols surrounded by a monolayer of phospholipids. They are coated with perilipin, which is a protein that acts as a protective coating from the body's natural lipases, such as hormone-sensitive lipase. When hormones such as epinephrine are secreted, or when insulin levels drop in response to low blood glucose level, this triggers an intracellular secondary messenger cascade that phosphorylates hormone-sensitive lipase to break triglycerides into glycerol and free fatty acids for use in metabolism in a process called lipolysis. The free fatty acids then move into the bloodstream, where they are bound by serum albumin and transported to the tissue needing fuel. Once the fatty acids reach the target tissue, they are released by serum albumin and cross into the cytosol of the mitochondria. The enzymes used in fatty acid oxidation in animal cells are located in the mitochondrial matrix. Free fatty acid chains of more than 12 carbons require the help of membrane transporters to cross into the membrane into the mitochondria, where they undergo fatty acid degradation. Fatty acid degradation is a process in which fatty acids are broken down, resulting in release of energy. It includes three major steps: activation and transporting to mitochondria, beta oxidation, and the electron transport chain, which we have discussed already under respiration. Fatty acids are transported across the outer mitochondrial membrane by carnitine acyl transferases, and then carried across the inner mitochondrial membrane by carnitine. Once inside the mitochondrial matrix, the fatty acyl carnitine reacts with coenzyme A to release the fatty acid and produce acetyl coenzyme A. CPT1 is believed to be the rate-limiting step in fatty acid oxidation. Once inside the mitochondrial matrix, fatty acids undergo beta oxidation. During this process, the two carbon molecules, acetyl coenzyme A, are repeatedly cleaved from the fatty acid. Acetyl coenzyme A can then enter the citric acid cycle, which produces NADH and FADH2, as described in Krebs cycle. NADH and FADH are subsequently used in the electron transport chain to produce ATP. The energy currency of the cell, since beta oxidation cleaves two carbon molecules repeatedly, it works well for even carbon chain length saturated fatty acids. So for odd carbon chain length fatty acid and unsaturated fatty acid, a slightly different pathways taken. Besides beta oxidation, other oxidative pathways are sometimes employed. Alpha oxidation is used for branched fatty acids that cannot directly undergo beta oxidation, such as phytanic acid. The smooth endoplasmic reticulum of the liver can perform that oxidation, which is primarily for detoxification, but can become much more prevalent in cases of defective beta oxidation. So fatty acids with very long chains, 20 or more, are first broken down to a manageable size in peroxisomes. In summary, the beta oxidation of fatty acids or lipolysis involves the following steps: dehydrogenation by acyl coenzyme A dehydrogenase yielding 1-FADH2, hydration by enol coenzyme A hydratase, hydrogenation by 3-hydroxyacyl coenzyme A dehydrogenase yielding 1-NADH, cleavage by thiolase yielding 1-acetyl coenzyme A and a fatty acid that has now been shortened by 2 carbons, acyl coenzyme A. This cycle repeats until the fatty acid has been completely reduced to acetyl coenzyme A. In the case of fatty acids with odd numbers of carbon atoms, acetyl coenzyme A and 1 molecule of propyl coenzyme A per molecule of fatty acid are produced after complete oxidation. The acetyl coenzyme A then goes through the Krebs cycle as described earlier. So in review, it's important that you should be able to state the sources of fatty acids, describe the composition and the digestion of fatty acids. Then the transport of fatty acids into the mitochondria was explained, and the beta oxidation of fatty acids was also pointed out. The session ended on the names of some enzymes involved in digestion, transport, and oxidation.